So Ritesh, I can record actually.
Yeah, I think I gave you the right. So,
Ach-
okay,
Okay. You go ahead.
okay. Um, Good afternoon, good morning, and good evening, wherever you are. Please welcome Vikranda, who's the speaker for today's Adda. He's a 1994 alumnus of Vidyapeeth. And the topic of today's Adda is the rise and rise of clinical gene. This Adda will be moderated by Amit Mathurda. He's a batchmate of Vikranda and from 1994. Uh, this is the first time ever that at least me, I'm face to face with both Amit and Vikrant as on video. And it's a great privilege to see two of our seniors who are going to interact. And it's a very important topic which I have no clue of. I hope the audience and the people who will watch the video later on will enjoy this. And if they have any further queries, they can contact Vikrant, and I will share his contact number on the chat box. Thank you very much. Amita, you can take over from this one.
Thanks, Ritesh. Welcome everyone. So welcome to this very interesting talk. So before Vikrant starts, let me just give you a very brief introduction. So Vikrant has a very interesting life story and very interesting academics, story. So Vikrant and I, we both passed out from Vidyapeeth, like Ritesh said, in 1994. After that, Vikrant did his +2 from Pilani and then undergraduation from Hansraj College, Delhi University. And he did his post-graduation from Institute of Genomics and Integrative Biology. And then he went on to do PhD from Indian Statistical Institute. I think that is where Vikrant's, mostly his life sciences journey starts. So he finished his PhD and then he moved to Singapore, where he is based right now. So for the past 20 years, Vikrant has been in Singapore. And in Singapore, he worked for various places, mainly Genome Institute of Singapore and then cancer and stem cell biology. But all throughout, he has been working in life sciences and mainly with molecular biology and genetics. Recently, basically right now, currently, Vikrant is working in a company based out of Singapore, which has been making waves all over the world. The company's name is Mirexis. And mostly Vikrant's work is even in Mirexis, it's focused on his same research area, which he will explain to us right now. It's mainly focused on life sciences and mainly focused on cancer research, using genetics. So it is an involved topic. It's a highly technical topic, but still the presentation is very interesting. To just set the baseline of what this presentation is about, Vikrant, I just want to ask you like some very basic question for most of us who are not so familiar with life sciences. Basically, my first question is, in layman's term, can you explain what is cancer?
Right. So cancer is all about a genetic change. Every cancer has to go through a genetic change. Without genetic change, there's no way, which leads to its immortality. The cell will never die and keeps on expanding and expanding. Each cell is programmed to die. That's called apoptosis. In fact, the person who proposed this apoptosis term got Nobel Prize some time back. And any cell which does not die is problematic.
Right.
Everybody
So
has to die
basically,
and it becomes.
yeah. So a cell which has been somehow by some mistake in the body, the cell has acquired a property that it will not die and it will keep multiplying.
Yeah.
Right. That is
And that
cancer.
property is a genetic change. That's cancer. That's in a
Right.
very
Right. So this
simple way.
whole
Yeah.
scary disease that we have heard about, various different types of cancer, ultimately every type of cancer, whatever it may be, lung cancer or blood cancer, it's ultimately derived from some genetic change that has happened within a cell in the body
Hmm.
so that it will not die and it will keep multiplying. Right?
That's true. That's true.
Right.
Yep.
Now, I think your research is mainly focused on how do we detect that somebody is prone to having some sort of a cancer? And then once somebody has a cancer, how to treat it? Is that right?
That's correct. I mean, one way is to figure it out early, much early in the stage, what kind of cancer they have. So one way is to focus on single cancer. Oh, I want to know whether I have got lung cancer or not. Let's say very highly smoking very good amount of cigarette.
Right.
The other way is that now there are products available in the market where you can screen at in one go many cancer through blood. Take the blood, figure out that you have a cancer. That's the first thing. Now, if you have cancer, where it is, and you have, it can be in the brain. Certainly, brain is difficult to figure out because there is a blood-brain barrier. But then you can have in lung, you can have in gastric, you can have colon, you can have anywhere,
Right.
prostate,
Right.
liver. The next question is where it is. Once you figure out these are the two things, you have cancer and where it is, then people can go for specific like PET technologies or whatever is available, or
Right.
scopy, colonoscopy or gastroscopy to confirm it, and then the
Right.
treatment start. Early, anything early is very important.
Correct.
Not only in
Correct.
cancer, it could be any other
Right.
disease.
Right. So it's basically a blood test. For the patient is just giving out a few drops of blood, using which they can tell you that you have early stage cancer, and then you go ahead with further tests to confirm it, right?
That's correct. That's correct.
And then once the cancer is found, I think your research also focuses on some ways of how we can treat it, again using the same genetic technologies, modifying the genes, etc.
Right. So actually, it's very important to have electronic electronic health record. You can take those electronic health record and look at the blood samples, and then this treatment was given in somebody, this treatment was given somebody, and then figure out retrospectively what mutation it was, and whether the treatment was successful. Then you can record it and see enrichment of those successful treatment based on a genetic
Right.
Otherwise, prospectively can be done as well, but it takes much more time. So both ways it can be done.
Right. Right.
Looking at the situation of
Okay. Very interesting. Very interesting. So basically, I will let you start, and
treatment
that
versus
is that
the kind
basically
of
gives us
genetic
some background.
alteration it has of cancer. Yes.
Thanks, Vikrant.
Yeah.
Okay. Why don't you start?
Right. So maybe, yeah. Yeah. Let me start this. Yeah. So then, okay. So very basic, very fundamental. So you have every has a sperm, every woman has egg, and those are so we all have 23 pairs. It's 22 pairs plus X and Y, then it's male. If it's 22 pairs X and X, it is female. Then every woman who has egg has only half the chromosome, which is 23, and every man has sperm, which is again 23. Once they fuse sperm and an egg, then a zygote forms. That zygote is what we are showing here. Let me just change my pointer. Yeah, then you have this fertilization happening, that zygote. And over a period of time, the zygote can become anything: stomach, pancreas, lung, muscles, and all those things. The cells keep on dividing. So all this stomach, pancreas, ultimately, they are all cells. Now, each cell has this 23 plus 23, 46 chromosome, which is this DNA double helix, Watson and Crick model. You can see that. And each cell will have this, except for the RBCs, red blood cell, which does not have. But otherwise, all the cells have in their nucleus DNA. Each cell has nucleus, and each nucleus has this chromosome, which is this structure. Now, what happens then? That DNA structure, as I said, each cell will have 46 human chromosome, and each chromosome has 2 copies except for X and Y. If it is X, it has 2 copies. If it is X and Y, it is X and Y, making it 46. 22 plus X and Y. That's making it 46. Now, the DNA, which is quite long, and they're packed very tightly. It's a 2 meters long DNA, and it is in very small, like even smaller than a picomole size. It's 2 meters of DNA. That has 3 billion DNA substitutes, or ATCGs. So it's a huge amount of alphabets. It's a 3 billion DNA, and they are in paired 80. So it is not one base, 3 into billion into 2 is actually 6 billion bases, but they always exist in pairs. Now, within these 3 billion bases, you have 30,000 genes which codes for proteins. So DNA leads to RNA, RNA leads to protein, and protein is the building block actually. And actually most of the drugs which targets are all proteins in the body. That's how it works. Now this 30,000 genes is only 2% or less than 2% of the entire genome, which is 3 billion bases. Rest of the 98% is very important. It helps in regulating these genes. So for example, if you have lung cells, so only certain genes will be expressed in lung, making it lung cells, very specialized for lung cells. Similarly, for heart, certain genes are going to be expressed, making it heart cells or cardiac cells. And those are that 98%. The 98% of the entire genome is basically helping you to express 30,000 genes selectively in different type of cell type, making it functional for each organ. To that part, if it is clear, I'm going to move to the next one. And please feel free to ask question or type. I would say maybe you can just ask question, stopping me and ask question. Now, what happens is that all fine, you have those kind of 3 billion bases, you have 20,000, 30,000 genes. Now, any mutation which is in the egg or in the sperm that is coming from your parents, that mutation will pass on onto the egg, which is a fusion of sperm and which passed on this zygote, fusion of sperm and egg, and then that mutation will be in your entire organism. In the entire, all the cells will carry that mutations, and that is we call it gametic or germline mutation. And I want to say that because it has a huge importance when we go further in the pathogenicity of these mutations. And then you have a somatic mutation where mutations is not acquired from sperm or egg, that means it is not inherited from your parents. These mutations occur later on in life. It can occur early on also, soon after the fertilization of sperm and egg, or it can occur any point of time. Those are called somatic mutations, and they are not passed on to your next generations unless the mutation happens in your own sperm or egg. Then you are passing into that. But that is a new mutations coming in sperm or egg. Usually somatic mutation will happen it can happen in sperm and egg though, but then those mutations will not pass into any generation unless it is sperm and egg. So, and both has his own importance. So very important aspects to distinguish between germline and somatic mutation. Now, germline mutation. So what happened was people were looking for this single change in one of the 3 billion bases, or that change can create all kind of very bad genetic diseases. Thankfully, the nature has created 2 copies, and most of the genes or most of the diseases, sorry, will require both the copies. I said each chromosome has 2 copies, that is 22, and then X and Y or an X and X, which is called sex chromosome. And each copy, if there are both the copies of mutation, then there is a big trouble. If these mutations are pathogenic mutation. Otherwise, a single copy doesn't really, your carrier, you can pass on to your kids, but then, or your kids can pass to their kids, but then it is not going to create problem for you because you're carrier. But the moment, let's say, a bad copy is passed from your father, another copy passed from your mother, then you have that problem. And those are called rare diseases. By and large, most of those rare diseases are 2 copies. Some of them are single copy also. That means if your parents, one of the parents have, you will certainly have it. 50% chance, if that mutation is there, you will have it. So initially, very few genes were picked up. Then what happened in 2000, this human genome draft came, that 3 billion bases human genome draft came. The entire 3 billion bases were read in a very meticulous way and a very painstakingly way. It took 10 years around, and $3 billion were kind of, the expenditure of having that 3 billion reading was $3 billion actually. And after that, huge acceleration happened to find out the mutation, because human genome draft gave you the entire, where are the genes, the entire annotations of the whole human genome. And by now, or let's say by 2018, we have around 4,000 genes known which will have mutations, monogenic disorders. This is very, very important to know those genes so that one can always screen in their parents. The parents can screen for before going for any kind of, if they want to have kids and all. I will come to that. On the other side, you have complex diseases, which is again based on all these germline mutations. For example, type 2 diabetes, which is very family oriented, like it is familial. Parents have, cousins have, there's a high likelihood you will have. Similarly, hypertension, blood pressure, family have it, you will see it has familial aggregation, or rheumatoid arthritis, or some of those other immune diseases like Crohn's diseases, ulcerative colitis. All these diseases have a familial aggregation, but they're not monogenic in the sense only one gene impacting, creating type 2 diabetes or something. No, it requires many, many genes coming together. And by, as of now, around 60,000 signals in that whole genome, we have figured it out. And you see, after 2000 draft of human genome, there was acceleration. Lot of things started coming up. And then over a period of time, by 2018, we have picked up, for example, blood pressure. There are more than 100 signals. And a subset of that signal can lead to blood pressure, hypertension. So these are typical cancer. They are heritable, but it is complex in nature. Monogenic are heritable in a very straight fashion. That's a difference. But all of them require germline mutation by and large, including some of the cancers like hereditary cancers as well. And then on the other side, you have somatic mutations, which leads to cancer. By and large, a lot of somatic mutations can lead to cancer, and it can impact any part of the body, brain, stomach, colorectal, breast, ovary, pancreas, you name it, and you will have it, except for lung, heart. Heart doesn't have usually any cancer as such, not known as, maybe very, very rare that it is not reported. But most of the organs will have cancer. And this somatic mutation, as I said earlier, you don't inherit it. It happens in one of the organs, and eventually you get it, and it's not there throughout the body. But some of the cancer could be because of germline. So cancer could be because of both, but somatic mutation is the most predominant way the cancer happens. And very interestingly, a new field has emerged, and it is emerging extremely fast. Somatic mutation in blood cells, leading to all kind of malignancy, cancer in the blood, hematological malignancies, heart problems, problems in the arteries, peripheral, or leading to heart problems because of the artery, hardening of the artery, diabetes, Alzheimer's, that means loss of memory in the brain, COPD, which is pulmonary disease of the lung diseases, liver diseases, chronic kidney diseases. And now papers are coming quick and fast. People are trying to understand what is changing in the blood cell, mutation in the blood cell, leading to all this kind of different kind of diseases. And most of the diseases are the diseases of aging. Heart disease you get much later in life. Diabetes also you don't get very early in life. Same goes for Alzheimer's. People always thought that it is nothing to do with mutations. Now it is more and more understood that it is to do with mutation. In fact, a mutation carrier of one of the mutation in blood, or overall mutation carrier in the blood, can lead to a 2 times risk of coronary heart and 4 times risk of early onset of myocardial infarction, which is another kind of, which is a heart disease. It's a kind of what we call heart attack. So all this can lead to cancer, other than cancer, can lead to other kind of disease as well. And it's very important to understand how can we use this information, because if I am a smoker, let's say, and I want to know, will I have a heart problem, or will I have some other kind of a problem, or COPD? Certainly smoking is one of the biggest risks, but I can always go to a lab and check out for those mutations by giving blood to screen for those mutations. If there is a mutation, let's say, I'm going to look for, I might go for MRI of my heart, or I'm going to look for a specific Alzheimer's disease. I'm going to look for MRI and look for plaques and all the stuff, whether it's happening or not. Because I just tell you, there are now a lot of antibodies available, monoclonal antibodies available, drugs are available for Alzheimer's. if it is picked up early, the treatment can be very good for Alzheimer's. The caveat is early. So all these mutations, if you can pick up early and you can inform the individual that you have a high chance, then you can go for MRI. Otherwise, going for MRI directly for all the individuals, it's very expensive, and there are not enough MRI to do all the individuals, and screening is going to be very, very expensive as such. So blood test becomes very easy to do. And if the blood test is positive, go for the next stage, which is a confirmatory test. And you are picking up early. This is the most important. Early is a very important term here. Then a treatment can start quickly and you have a much better result. And I will show you some examples in cancer, actually how it works. Okay, so all these questions, now you know germline can create all kind of mutations, somatic can create all kind of mutations. What one can do with this kind of information, how it can help in prevention. Actually, preventing healthcare is becoming the most important aspect nowadays, and precision healthcare, which is after you figure it out what kind of problem you have, then what kind of treatment should be given. And this is all informed by kind of all the mutations you have, all kind of genetic changes you have. And also I wanted to show you that first time ever the COVID pandemic came, the first vaccine ever was an RNA vaccine. No ever the vaccine was an RNA vaccine. It was actually repurposed. The vaccine was supposed to be for cancer. That biotech company in Germany, they repurposed this quickly to come out with a vaccine for COVID, which did a wonderful job. And then it was sold by Pfizer. And then, of course, then Moderna came as well. So both these companies used the RNA method to create vaccine, and it was a wonderful vaccine, actually. Then I'm going to show how, for example, if you have a hereditary like the germline, what you can do. And if you have somatic, what you can do, how can you figure out? And then if you have to figure out, okay, you have this kind of mutations, then what kind of therapy is required? That is very important. That's the ultimate aspect of it. Let's look at heredity, for example. Now, if I look at Singapore 10,000 samples, whole genome was sequenced, all the entire 30,000 gene, in fact, entire 3 billion bases were sequenced. And you can see a lot of diseases are there in Chinese cohort, Indian cohorts, Malay cohort. For example, cystic fibrosis, which is a bad disease, it creates all kind of mucus in the lung and all. And the frequency is very high in Chinese. Almost 8% of the individual can have it if this one in a normal population. Now, if I know the mutation, right now there is no treatment as such if you have early picked up, but then people are working on that. Can we pick up early and start treating on that? So that's a very important aspect of it. People having beta thalassemia, can we pick it up and start treating it early, or can give us blood transfusion early so that longevity can also increase, and all the stuff can happen if you are picked up. So two aspects happen. One is to know what kind of problem I have, what is the genetic basis, and second, the treatment aspect of it. So this is very important. And the third more important thing is that if I am a carrier of it, I may not have the disease because I'm not carrying two copies, and my wife also has it, one of the copies, then my kid can have it. So we would like to go for screening before any kind of pregnancy to happen, before we decide to have kids. So this kind of screening can happen. So for example, there is a panel already existing where you can pick up 421 disorders. I just showed a glimpse of it. On the male, you can pick up 381 disorders. And you can ask why this so much, because there is an X chromosome involved on the female that increases your disorder numbers. And all this can be screened. The question is, when do you go for that? Couple trying to conceive or expanding their family, all pregnant women, as well as sometimes you go for in vitro fertilization and all the stuff. There also they have it. Now, if you are a healthy carrier, father, and the mother is also a healthy carrier, the chances that baby will be affected is 25%, is a very high chance. So it's better to go for and check it, and then you can decide whether you want to have that, or you can decide that, okay, I would like to have that baby, but I will go for, now there is something called, I don't want to go into details, called pre-seq. I can take the mother's blood, and from the mother's blood, the pregnant mother's blood, I can, based on mother's blood, I can screen and I can find out whether my baby is affected or not. So that technology has gone to that level that I'm a healthy career father, I'm a healthy career mother, but both are carrier, and my baby is affected or not. I can do after conceiving also. The mother can do after conceiving, because mother's blood can show, because it is that the technology allows you to pick up small snippets of baby's DNA in mother's blood. You can read those DNA and can say, oh, the baby is affected, and then you can decide to terminate or not terminate. All this can happen. So it's very important to go for this kind of testing for couples who are carriers, both of them. And to know whether they are carrier or not, you have to anyway do this, what kind of testing I just said. Now, going to another aspect, which is hereditary cancer testing. Approximately 1 in 6 people in Southeast Asia will be affected by cancer during their lifetime. And hereditary cancer occurs in 5 to 10% of those cases and are caused by inherited, as I said, pathogenic germline mutation in one or more genes. And right now, there are panels. For example, there is a panel which can analyze 94 genes in one go and can inform whether one has inherited that cancer from one of the parents. It could be father or mother, and then decide whether I would like to have. Based on that, they can decide what you would like to do that. So all cancers are genetic and, as I said, and caused by changes in genes that control the way cell function. And there are random mistakes in DNA. It could be carcinogen in environment, which are leading to changes in the DNA. All carcinogen will lead to DNA changes. And it could be inherited from parents. I said 5 to 10%. So sporadic are the somatic ones. 5 to 10% we know. There are still 15 to 20% cases we do not know. They are familial. If it's in the family, but you do not know which gene it is causing. So we're still struggling to find out what's happening there. Now, if you have mutations, these are the genes, it's high probability that if the father carries it, father will not have those, let's say BRCA1, BRCA2, leading to breast cancer or ovarian cancer. The father will not have, but if it passes to the girl has a high probability, almost 80% chance that the girl will have it. And if the mother has it, the mother will have it later in the life, and it will pass to the kid, the girl kid will have it. So it's important to know what it is. As I said here, it is a dominant mode, means that if the mother has it and it passes to the kid will also have it. That's a dominant mode. It doesn't require 2 copies, it requires just 1 copy. And then the risk is very, very high that if you have 1 copy, you will have it. So people tend to remove their breast very early or uterus very early so that they do not have it, because it is very aggressive. This cancer is extremely aggressive, so they remove it. So it's important to know this kind of genes. And there are other genes as well. I've not listed those genes out. If you have that mutation, it's better to remove those organs and then live a good life after that. And I have that actually. This is a gene compressive hereditary cancer test. A lot of them are here. One can look at it and decide based on what it is. Now, this is again picking up early. As I said, you are carrier and you are passing to your kids or not, you can figure it out very early. The other aspect is early detection of some of those cancer, or as I said, for example, Alzheimer's. I will give you this good example of gastroclear. It's a gastric cancer which used to be very predominant in Southeast Asia, in Japan, in China, in Singapore, and on the whole of Southeast Asia actually. But the gastric cancer, by and large, majority of the patients were diagnosed at stage 3 and stage 4. You have no symptoms in stage 1 and stage 2. Or very few people have symptoms that you will go to a doctor and say, hey, I have problem in my stomach, and then doctor will go for gastroscopy, colonoscopy, or any kind of scopy. It's better. Doctor will say, and by the time doctor sees that, and the doctor says, oh, you have on stage 3 and stage 4, and then basically doctor tries to manage your a good life rather than treating it. Tries to treat it, but then survival rates are the 5-year survival is very, very low. For example, if you look at 5-year survival of gastric cancer, stage 3 and stage 4, number of people surviving 5 years is very low. 5-year survival rate is on your y-axis, and these are the different stages. For example, stage 4, only 5% of the people are around which will live for 5 years. Or stage 3, again, less than 10% of the people, maybe less than 20% of the people are going to live for 5 years. But if you picked up stage 1 and stage 2, you're going to live for a long life. That's the important of it. Now, people who have rolled out gastric cancer screening, like in Korea and Japan, the 5-year survival rate has increased significantly, almost 70% in Korea and 60% in Japan. People where it is not rolled out in China, US, and Malaysia, the percentage is very, very low of 5-year survival. So for example, this GastroClear, you give the blood and then genetic testing will be done. Even if you have no cancer, pre-stage, that's called high-grade dysplasia, you can be picked up 60% of the time correctly that you have going to have cancer. And if you have stage 1, almost 87% you can be picked up, stage 2, 90% you can be picked up. So this is what one has to focus on. Can you pick up early, and then the treatment can be followed. So for example, what happens? Who will go for early screening? People who have in their family history of stomach cancer, you have history of stomach problem like gastritis, you have H. pylori, or you are obese, lifestyle problem, tobacco, alcohol. These are the things. If you have, you go for it. They're just scoring. If you have a high score, then go for a gastric. Go to a gastroenterologist, and then they will follow it with a gastroscopy. And that's how it works very nicely. If you have a low score, don't worry. If you have intermediate score, then you go for screening again after 6 months or 1 year, and then look for it whether you will have it, or you can even go for gastroscopy. It's up to the recommendation of the doctor based on the other clinical history of the person. So if you are picked up early, then what happens? Then you want to know, okay, I picked up early. What kind of therapy has to be given? And that's where this kind of therapy selection happens. So this is one COMPASS comprehensive therapy, which is 1,000 genes, all kind of. So gastrointestinal is there, lung cancer is there, breast cancer, cholangiocarcinoma is a liver cancer, one kind of liver cancer, colorectal cancer, endometrial cancer, ovarian cancer, and all other kind of solid tumors are also there, where based on the alteration in the gene, the doctor can decide what kind of treatment should be given. In fact, I will tell you one very interesting story about it. So that 1,000 genes, as I said, based on that 1,000 gene, any change in the mutation, so sorry, I think I will answer this question later, Somesh. I just saw it now. There are 97 FDA approved therapies already available. So depending on what treatment one can get from these therapies, there are 63 trials in a very advanced stage, clinical stage 3 already there, which people are allowed to use one of those because they have already crossed the toxicity safety. And then there are chemotherapies also available. And very interestingly, which is very expensive, called immune checkpoint inhibitor, ICIs, or which comes under the class of immunotherapy, also available. It's, as I said, it's very expensive, but then if you have a very high level of this indication, what called tumor mutational burden or MSI, this therapy works wonderful. Only if you have very high percentage. And then because these are very expensive, and if these indications are not there, this expensive ones doesn't work. So you lose your time to go for next line of treatment. And these therapies are expensive, you're losing a lot of money. So these genes help you at the first line or second line of treatment as sometimes people go for second line, but now the cost of using this kind of panels have really gone down the cost. So you can actually go in the first line of therapy and tell doctor that I want to use this particular panel to find out which therapy I should take, and which makes things very, oh, what happened? This is not going, yeah. So for example, there are some of the mutations. I was just showing you lung and breast is very common mutation. So if somebody has an EGFR mutation, I don't want to go into L, what is L858R, and it is positive for that mutation, there's a treatment available. And this treatment works very good. On the other hand, if you have a mutation, then certainly you cannot be given this treatment. It's a contraindication. You cannot be given this treatment. So doctor knows if you have a mutation, which treatment should not be given, because of that mutation, the cancer is resistant to this treatment. So doctor has to know which therapy to be given and which therapy need not be given. Very important. So this is how doctors figures out what therapy should go and what therapy should not be given. And I picked up lung and breast because they are most common cancer around the world. In female, it is breast, the highest incidence. In men, it is lung. And EGFR, as I said, is a very important gene, extremely important gene. So is HER2. In fact, EGFR and HER2 now has become very, very common. And in melanoma, this is a skin cancer. BRF is another very, very important gene. And this kind of treatment is informed by the gene mutations. Now, how doctors figure it out? Patient with advanced solid tumor, let's say you are already in stage 3 and stage 4, still go for this panel. Of course, if you have picked up early stage 1, stage 2, because you had a way to pick it up, undoubtedly you should go for it. And then first, second line treatment for sure, because it is not cheap, but now it has become very, very cheap. So people go for first line treatment. And then you have, again, personalized treatment planning, single test can happen. Let's say your first line treatment didn't work. Then doctor sees what other genetic alterations are there. They can go for second line of treatment based on the same panel, which was already done. So this kind of things helps a lot. As I said, immunotherapy selections has become very important as well. Those TMB, as I said, MSI, I said, other one is PDL1, which I did not show there. They're very important at what kind of immune checkpoint inhibitors can be given. And these are new class of treatment, very powerful treatment, good treatment, but the question is when to be given, and these are the way to figure it out. And lastly, I think I've probably gone beyond my remit. I'm going to stop there. Now there are gene therapies coming up. For example, 2 therapies are already FDA approved, one for sickle cell trait, another is for hemophilia. More and more are coming, and they are using CRISPR, where the gene can be edited directly either in the system of the human being or this, which I'm going to explain a little bit. And this is another way of treating. In fact, this technology, which is called CRISPR, which allows the gene to be manipulated, it is a very recent technology, and in fact, but the technology is so powerful that people who came out with this technology, they were given Nobel Prize very recently as well. So it's very interesting that a technology which is a very recent technology and Nobel Prize is given soon after. Usually it doesn't happen. It takes like at least few decades before a Nobel Prize is given. But in this case, the technology was very powerful. I give you a very important example on cancer, let's say. There's this kind of one, this is a T cell, a kind of immune cell in blood. You can take the T cell out from the blood. And then using this CRISPR technology, you can insert a gene. In this case, they have inserted a gene called NY-ESO. I don't want to go into details of it. And then you can remove certain genes called PDC1 and this kind of genes. So you have added a gene and you have removed a gene in that 3 billion bases. After adding and after removing, that is manipulating the DNA, you grow that in the lab, that cell. And this is your own cell. So it's very good that it's your own cell. Otherwise, this problem which happens when you take, when organ transplant happen, you have all kind of problems. This kind of problems can be taken care of if you have your own cell. And then you harvest that cell or grow that cell. After growing, you harvest the cell, and then you infuse it in the body, and it will very nicely go and kill the cancer cell. And of course, it will kill some other cells as well, the normal cells, but it is preferentially going to kill cancer cell. And that's how it is. But these technologies are not cheap. It's coming up fast. For example, in India, this technology is somebody has launched for a particular cancer type, and they say it's around 30 to 40 lakhs. Outside it is a $500,000 is the pricing, but this technology is really, really good. People are working hard on it to make it so that adverse effects are not there, and also the cost can come down so that it is available for all the people. I'm going to stop here, and I'm going to say thank you to you guys for attending this and listening to me patiently. Yeah, I'm going to stop and then take questions. Thank you very much.
Thanks, Vikrant. Thank you. So there are two questions so far for you. Others, please ask or type on the chat. So Vikrant, the first question is, for the blood test to screen for cancer, what is the typical expense?
Okay, that's a good question. Right now, you can bargain. For example, I give you one example. When my wife was pregnant, we wanted to go for an IPT called non-invasive prenatal test, where my wife's blood was taken, and I'm talking about 10, 12 years back. And you can pick up some of those Down syndrome, Edwards syndrome, and all the stuff using mother's blood. And it was $2,000 that point of time. And before we went, it was $3,000. And we said, okay, we'll come after a week or so. We'll think about it. And then the clinic calls me and say, hey, the lab is decided to give you a $500 discount because they wanted to penetrate the market. You have to be smart in bargaining that. It can really, really go down. Now, for example, GastroClear, for the cancer screening, it is somewhere around $500, $600. You can bargain. It's a lot of bargaining happening right now,
Right.
so that it can be penetrated in the market. Because the awareness is low, the cost is high. As the awareness will be more and more, and we know in the lab it is not very expensive. It's just that the cost is high because the awareness is low. So and then as more and more will be picked up, the cost will come down. That's one thing. Yes.
Right. Next question for you is that for stomach cancer, where can we do the test in India? Is it available in India anywhere?
Not for stomach cancer right now. In Southeast Asia, yes. And this GastroClear is gone for FDA. It has been adopted by FDA accelerated program, which means they pick up some of those tests which they see a strong value for the population, and they try to clear it within 1 or 2 years. Otherwise, FDA takes their 5 years to 10 years time on an average. So we have been able to get into their accelerated program, and we are hoping that but then Japan has allowed it, Singapore has allowed it, their equivalent to FDA, they've allowed it. We have not gone into India. As I said, I was telling you in the afternoon that we're trying to see whether we should go India or not. There is one comprehensive already launched by one company in the US, which can do 32 cancer early detection in one go, just using blood.
In UK, in the UK.
No, it's a US based company.
Okay.
And the product is called Galeria, but it may not be cheap. Just to let you know.
Right. How much roughly? So basically, if somebody desperately wants it, they have to travel to US. But how much will that cost?
You never know. It's there in India. It's just that I don't know. But it might cost you somewhere around. I mean, somewhere below 10K, I would say. But again, I do not know how much it will be in India. Unfortunately,
$10K.
Should be 5 to 6K here. Yeah, 5 to 6K.
5
in
to
India,
6K.
again, India, they should give a cheaper one. So, but again, I don't know how much it will be. People in India might know. So, it's a 32, 34 solid cancer. It will not include brain for sure. I know brain cancer, but then it's a very comprehensive panel, and it can tell you with a very great precision whether you have a cancer or not in stage 1 or 2, or even 3 or 4, and you have no symptoms, let's say. Or you might get symptoms 6 months later. And at the same time, which organ has that? So that's where, and it's important to know also. Once you know, then you go and look at that organ using PET technology or whatever means, or scopy. Then you take a biopsy, and then you figure out what is the genetic alteration, and then you go for, based on that alteration, you go for a therapy. I'll give you a very interesting. Let me tell you a story. Some few years back, when it was very expensive, sequencing was too expensive. So there was this guy in Broad Institute. Broad Institute is a collaboration of MIT and Harvard in Boston to look for, and it's a marker for genetic work. And there was this guy who was working in cancer, and this guy was extremely good in. He was fitness freak, but unfortunately, very young stage, at like 30 years old, he developed cancer. I mean, he was feeling fatigue and all, and and he found out that he has cancer, and it was leukemia. And I'm not going to detail of what kind of leukemia and all the stuff. And then he was like, wow, leukemia, then what to do? And then he knew that some of the leukemia, if you do a whole genome sequencing or all the gene sequencing, 30,000 genes, I might find out the change, and then I can find out what treatment is available. But then, as I said, it was very expensive. So the lab did crowdfunding for that person. And then sequencing was done, and they found out the mutation. They found out the mutation, but that mutation was indication in liver cancer. But they found out in leukemia also. And because of that mutation in liver, there was a treatment available off the shelf. That means you don't have to go and go for experimental and all. It was available. But the insurance company said, we will not pay you for this treatment because it is only allowed for liver cancer.
Right.
And that guy wrote to Pfizer also, but the Pfizer also denied, and that therapy was very expensive. But anyhow, he went ahead and took that particular treatment without insurance, and in 2 months' time, his cancer was clear.
Wow.
Or leukemia. So cancer is no more now organ specific. It has become gene alteration specific. Things are changing. It is becoming very molecular now. We used to say earlier, oh, he has liver cancer. Now EGFR, one of the mutation in lung, you can have it in gastric, you can have in colorectal, you can have anywhere, and same treatment will be allowed.
Right.
Which was on lung, another, and Pfizer quickly saw that opportunity and gave him free and made it an indication for leukemia as well. So this is how companies work as well. Yeah.
Right. Right.
So
Very
somebody has
interesting.
asked, So somebody has asked, if somebody's parents were diagnosed with stomach cancer, what test should they undergo and where in India can they run? Unfortunately, I don't know, but they should certainly go for some kind of a test, whether it's HER2 test, EGFR test. These are very important. There are some companies who do one gene test as well. You must go for a test, and that will tell you what kind of therapy. Test is very important. For example, Singapore is the largest hospital, National University Hospital. All their samples comes to us. We have a joint venture. All their samples comes to us to tell them which treatment to be given. So now none of them go for just any kind of treatment. No, they are going for treatments based on information on the gene alteration. Unfortunately, I do not know. So what kind of sequencing tech you use? Illumina. Solid has folded up. There's nothing. There are few now benchtop of Illumina as well. There is MGI platform, which is a platform from China. There is companies like coming from Thermo Fisher. There are few which will allow you to do sequencing. And you don't have to do sequencing. Sometimes you can do other ways also. You can detect it. Something we call, everybody now know PCR because of COVID. There's something called qPCR, quantitative PCR, that can also tell you what kind of mutation is there if you are looking for single mutations. Somesh has asked, what test is that a person should do to help early identification of any threat? Yes, Somesh, it is little expensive. Doctors are not aware of it. As I was there recently in India, most of the doctors, including cancer doctors, don't know there's a test available. India is somehow, and also, I don't blame them. It's just that the education curriculum needs to change in a wholesale manner to figure it out, these things. Yeah. So yeah, unfortunately, that's not happening right now in India, but I'm hoping in next 5 to 10 years, more and more people are aware of this kind of testing. It's just a blood test. So most of the tests are actually meant for blood tests. And now very recently, the paper came out in JAMA Neurology. JAMA is a Journal of American Medical Association, where with 90% accuracy with blood test, you can pick up Alzheimer's. Now there are 3 drugs already available in Alzheimer's. If you are Alzheimer's picked up early, you can really make it progress very slow. You won't stop it, but so slow that you might die of some other disease rather than Alzheimer's. But the trick is to pick up early.
Right.
So, yeah, so blood has certain kind of protein. That's a protein-based test. It's a combination of 2 proteins. Now the question is, you can ask when India will take it up. Well, question is when people will roll it out in India. I mean, even immune checkpoint inhibition therapy came in India very late. CAR-Ts are not even available in India, where T cells are taken out of the body and then manipulate and then give it back. Hardly. There's one company, I think that company, yeah, I saw the news that they can do it for 30 to 40 lakhs, but these are the things where you can do therapies. So therapies are a problem in India nowadays. The identification is a problem in India nowadays. And also people are not aware of it, so that they can go and ask their doctor, why can you launch it? Yeah, so unfortunately, these are the things.
Is there any age cutoff, after which people should try to screen themselves, do like a, or can it happen at any age?
It's a good question. I mean, so most of this disease of cancer, of heart, usually doesn't happen in early. So you are 40 plus, you are on the wrong side. Evolutionarily speaking, people were not meant to live so long. You have given birth to your babies, you are good to die. Because procreation is the need, speciation is the way. Now infection has gone, most of this is taken care of. Now cells have to really work hard to keep you alive. Uh, there is a new paradigm which is picking up very fast, and as I said, blood one. This mutations in the blood is all full of immune cells, full of all immune cells, right? These immune cells, blood, when they have mutation, they go and start attacking their own cells in heart, they start attacking in the muscles, leading to all kind of aging problem in brain as well. Um, so so now a new impetus is coming that at least in Singapore, there is a national initiative to map out all these blood mutations with aging and all the clinical whatever parameters are there, because it's easy to access electronic health records are very well maintained in Singapore, extremely well maintained. The correlations can be done. And then once you know what are the treatment, once you know I have this mutation in immune cell, the first thing I will look at my heart. Second thing I will look at, I will be very cautious about cancer of the blood. And third is kidney. These are the major ones I will be always looking at. Because kidney failures are becoming more and more as you age, same as the heart. And a lot of them to do with smoking is certainly one big problem, but then it will be compounded by these mutations. Also, it will be without that, without smoking also sometimes you will have it. So sometimes you ask, some people are smoking and they die, some people are smoking, they don't die. It's a combination of many things as well. But smoking as such is a very, very important factor because it changes DNA. Nicotine can change DNA. That's why it is carcinogen.
Right. Right.
And it's important to see that nicotine is, and first time when the cigarette or smoking was made, was picked up as one of the problem for cancer. He was an epidemiologist in UK. He was one of the chief epidemiologists UK. He was put into trial, to taken to the court by big companies, all these big cigarette companies, that he's telling lies, smoking has nothing to do with it, is a lifestyle you enjoy and all the stuff. So that's how science progressed, you see. And now that guy became a big guy at some point of time, bestowed with some kind of, he was given this sirhood and all the stuff in UK. So yeah, so the story is one should be aware of. In India, things are not that great. I think because people have other priorities as well. I mean, your tuberculosis as such is another big problem which is not being taken care of. that's infectious disease. Forget about cancer and the stuff. Infection has to be taken care of. Tuberculosis, malaria, and other stuff. AIDS, including, of course, AIDS has come down because of those drugs, but problem persists in India. It has its own set of problems. So difficult to answer these questions. But if you can afford it, go for this kind of test. And if this test say yes, then certainly go for, depending on what doctor says, CT scan, MRI, scopies, or any kind of things. It is very helpful. Yeah. And we
Anybody
all,
else has any other question for Vikrant? We are almost out of time.
yeah, we
Yeah.
are quite a bit.
Yeah, may I ask a couple of questions? One is, some of these seem to be very specific. So, for example, some tests targeting disorders, carcinogenic disorders of the stomach, for example. But is there any comprehensive test which will give you an idea of whatever genetic mutations you may have? Because from there, one may proceed to look for potential, possibilities of things cropping up later. This is question one. The second is there any diagnostic role that these tests can play? So, for example, somebody has a tumor, and there is a problem in diagnosing what kind of, even histopathology is not able to tell you whether it is malignant. Can genomic testing help? And finally, is it not possible to send samples abroad to get these tests done if they are not available in India?
Yeah, last question first. You can, as long as it is clinical sample, you are allowed to send it outside India. Non-clinical samples, things are becoming difficult. Government is becoming, especially because of genetics, every country is not allowing, except for the Western world, to send you samples for doing genetic sequencing. Every country wants to kind of becoming sovereign. They want to keep their data in their own country, particularly for genes. I do not know. This is ridiculous. I don't like that. Most of the researchers don't like that. Why data should be held at each country? China particularly does not allow any sample to go out, including clinical. But at least in India, clinical samples can go out. You can look for the test. Secondly, if the cancer is malignant, histopathologists should be able to pick it up. It's very straightforward. If you're doing HNE stain, you should be able to pick it up. That's the gold standard. If you're not able to pick it up, then either the cells, they have picked up a wrong biopsy. That's the thing. And the question is why the biopsy happened. If the histopathologist there was some kind of symptoms, right? So you picked it up, and if it is not, most likely it is benign. Nothing is there. It's highly likely that it's unlikely that it's a cancer. To your first question, in terms of genetic comprehensive test, yes, as I said, there is a company which has launched called Galleria for 34 cancer test, 32 cancer test. I don't know exactly number of solid tumor. It is possible that out of 32, they will say, oh, I will only tell you for 5 or 10. If you give me $1,000, if you give me $10,000, I will tell you for all 32. I do not know their working style, their model, but that can do up to 32 solid cancer tests. So yes, you can send it outside, maybe US and all, and then they have clinical labs all over the world, and they can extract whatever is required to be extracted. They will use plasma actually from the blood, and then from plasma they will take the DNA, and then they will look into what is there. Those are small fragments of DNA and plasma floating around. Yes, so that can be done, but that test is a different kind of a test. It's not mutation test. It's a epigenetic test. It's the way genes regulate the expression of genes. So those regulatory tests are there. So in fact, the gastroclear is also not a alteration of gene. You're looking at something beyond that. How genes are expressed, That's how GastroClear. We are soon coming out. We have just launched lung cancer test as well, because lung is another big problem for the world. We just launched a few months back in Singapore, and then we launch all around the world. Early detection of cancer. People have launched for Alzheimer's. People have launched for another kind of test where what happens is that you have been given chemo or you have been given, and then doctor says, oh, perfectly all right. You are into remission. There is no nothing. You don't see any kind of neoplasm or any kind of tissue which is cancerous. All good. And then after 2-3 years, you go for PET because, and doctor says, hey, your cancer is back. And then usually it is very late. So we call it MRD, minimum residual disease. So the trick is PET can pick up cancer, but which is the gold standard, but it picks up very late. However, picking it very early. So again, the cancer starts developing any part of the body after metastasis. That means it's moving all around the body. So even before that, once the cancer again starts growing, it will again start shedding the cancer's DNA in the blood. And again, using blood, you can pick it up. If you can pick it up in, let's say, first 2 to 3 months after the cancer has started again, the treatment works very well. So which we call that you have relapsed, but then you picked up much earlier, almost 5-6 months earlier. PET can pick up like 7, 8, 9, 10 months after the relapse. And that's a huge time you have lost. If it can be picked up very early, 2-3 months after the relapse, or let's say you've gone for surgery was not successful, doctor sent you back, and after 2-3 months you go and you find out cancer is there, but that PET cannot pick it up. Only that blood test can pick it up. So that kind of blood test is also available, but those are not cheap and they're very sensitive. It's not easy to do that, but there are companies like Lucin is one company, which is a Singapore company. Now it is probably going to be listed in the US. So these are the tests which is available. Again, awareness is very low, extremely low. We keep on teaching doctors all the time. So when I was in Duke Medical School, I was taking some of the things for the students there. So and we realized that more and more curriculum has to change to bring in all this kind of information, how genes are changing things, how there is a paradigm shift in most of the diseases, especially late onset ones, because these are the disease of aging, and a lot of them is governed by genes and changes in genes actually. Yep.
Is Galeria available in Singapore?
Galeria, I don't know. Should be available in Singapore, or I can send it outside to US. I don't know.
Any idea what it costs?
I would think it's around less than 10K or 5-6K. I mean, it's very early. It has launched, so I'm pretty sure you have the power to bargain. As simple as that.
Well,
say, no. I don't want to go for this. I have to. They say no, it's too much. They'll come back to you because they want to increase the number of samples and they want to penetrate the market, so they might give it cheaper one. I do not know the cost, unfortunately, sir. If I come to know, I'll let you know.
I was just hoping that you could do the bargaining for me.
No.
Because obviously you're much more knowledgeable.
Right. So no, Galeria, I have not. In fact, but then I've enrolled in one of the programs in Singapore where they have taken all my stats of CT scan, MRI, every part of the organ, everything they have done. And they have taken blood and they're going to follow me for 4 years. Every year I have to give them blood. And they are going to look all the mutations in the blood particularly and the heart and correlate with my clinical aspects. And there is a huge, and there will be 20,000 samples. And Indians are obviously very low here. So it was very easy for me to get registered in that. So these kind of programs are there at national level in Singapore. It's a small country, can be done. India is a huge country, so it has the advantage of the sample size, but then there's nobody doing it, unfortunately. The cohorts are very, very low in number, or the clinical data set is very sparse or not very well managed or taken. So there are problems in India, unfortunately. And I think Amit's actually Mathu, which the company is, they probably have a much better source of getting clinical data and sample perhaps. And that, I tell you one example of UK Biobank. UK Biobank did sequencing of 500,000 sample. 500,000, that's a huge number.
Mhm.
And what they did was they asked companies to come, that I'll give you the genetic information, I will give you the clinical information. Companies like AstraZeneca, Pfizer, Amgen, all big companies, Novartis, they said, okay, we look at the proteins and then see which protein is impacted by the genes. And it was a wealth information. They picked up all the proteins which is directly governed by the gene, changes in the gene, changing in the protein expression, and then now they have that information. They will go to the lab and start making drugs for each of the protein. So I have that genetic mutation, pathogenic genetic mutation, changing the protein, then drug will come, attack that protein. It was a cool way of UK Biobank, but then UK Biobank did not come in 1 year. It was a huge effort of lot many people, and then 500,000 samples. That's a lot of samples, which, and in India is huge, 1.2 or 1.4 billion. So sample size is never going to be a problem. It's a data, it's a how to use the human capital of India. But yeah, that's how
Finally,
it is.
will you please share your email on the chat so that we can get in touch?
Sure, I can do that.
Thank you.
I mean, email, I'm less likely I will answer, but my phone's WhatsApp, I would.
Okay,
Phone is,
that's fine.
yeah,
That's
and this
fine.
number doesn't change actually. So yeah, unless until I change the country. Yeah, so yeah, this is what it is. Unfortunately, India is not on the cusp of, India is in the cusp of, not cusp, India has already become uh, a pandemic, I would say, of diabetes, of hypertension, heart problems, and soon kidney. Nobody is realizing that, and I think government is realizing that. Just that government doesn't have the resources to manage things. Unfortunately, it's a lot of problems India will face because the longevity is increasing in India very fast, and these are problems of aging. So frailty, which is muscle weakening, all of the stuff, and dementia. They're the two major big problems India is going to face soon. Not going to take much time. Soon. So yeah, and I do not know how the government is going to work on these aspects of frailty and dementia. The research R&D, if you see the last 10 years particularly, I'm not a BJP or Congress supporter, but I was just looking at one of the data on the research R&D, the GDP to the ratio of R&D expenditure on the GDP side, the budget, it has been diminishing every year, and which is a sad news.
Right.
It's a clear trend. I do not know what has happened. It has happened in the Congress time also, but somehow it was a later part of Manmohan Singh, they increased the R&D budget, so that linear trend was not there. But here there is a clear trend. But that's how it is.
And thank you, Vikrant.
Yep.
Thank you, Vikrant. Thanks a lot.
Thank you, guys.
Thanks a
Thank
lot
you.
for spending
Thanks
the
for listening
time
me
with
up.
us and explaining us, all these difficult topics so nicely. Thanks.
Thank you. Thank you, guys, for listening.